So first, thanks a lot to the organizers for inviting me to speak on the statistical challenges with master protocols. And as mentioned by Dieter before, I'm involved in the EU Pearl project. And so most of the work I show today is coming out of this project, which is an MI-funded project, which shall develop a framework on how to design platform trials, and it includes several working groups on statistical topics such as multiplicity and use of non-concurrent control data. And it's funded by MI, so the Innovative Medicines Initiative. You can see the acknowledgement here on this slide. And there we also have several indications, like broad indications like NASH or depression, but also some in the rare disease setting like neurofibromatosis, because the purpose is really to develop a joint framework on platform trials. So here you can see the main issues I will talk about. First, I will set the scene again. And from the statistical point of view, I will mainly focus on multiplicity issues as presented by Daniel now, and also about which data we can use in the analysis. And if there's some time left, I might also reflect on the role of clinical trial simulations when we set up such more complex trial designs. Good. So let's first look at classical drug development programs. And before master protocols were introduced, how have most drug development programs looked like, and how does it relate to multiplicity and sharing of controls? There might be many studies in parallel, but each trial stands on its own feet, each trial which is a unique design, and each has its own control, and there's no sharing of data across trials. So here you could argue, traditionally, we might have learned a lot about the controls, and also traditionally, we would focus on type 1 error control at study level, which is also pointed out in several regulatory guidelines, like in the ICH 9 or on the one of multiple testing issued by the FDA or EMA. But the key point is really we don't share information across studies and indication. And now, as it worked for many, many decades, you might wonder, why was there a wish for something different? Because it might be sometimes an inefficient use of resources, because each standalone RCD needs its own control group, and every time we have to invent the wheel again and again, because we have to develop a new protocol, a new SAP. For each trial, we have to seek ethical approval, regulatory approval. We might have to look for appropriate trial sites. So it costs a lot of efforts to start a new trial. And therefore, some smart people came up with the idea of master protocol. And if you haven't read it yet, you might go back to the fundamental paper by Woodcock and Lavange. And I will now introduce the concept of basket, umbrella, and platform trials using their terminology. So what are basket trials? So when we speak about basket trials, it means I'm interested in a single investigational treatment, but this treatment is evaluated in the context of multiple diseases or disease subtypes with a common therapeutic target. So I have one treatment and I tried in a various in multiple diseases. Then we have umbrella trials. Now it's vice versa. Here we have multiple investigational treatments, which are evaluated in the context of a single disease, possibly within several sub-studies for different disease subtypes. And the combination of everything are platform trials. You could also say this is umbrella trials, where we test drugs or combinations thereof, which can enter and leave the trial at any point in time. So they are really quite flexible. And most parts of my presentation will focus on platform trials. Good. So first, let's start with a literature review. So there we looked which type of master protocols are actually executed and in which phase of drug development and which analysis methods are actually used. It was published, I think, 2020. So you might say it's a little bit outdated, and you might be right, because especially due to the last 3 years of pandemic, there was a huge push for this type of trial designs in the context of COVID trials. So now the review might look a little bit differently, but still it might give us a good impression on what's ongoing. So first, if you can see, here you can see that platform trials are mainly applied in the early phases, mainly in phase 2. So most of the trials conducted are in phase 2. And this is in contrast what we have seen in the survey, where most people say the highest potential might be for a combined phase 2B and phase 3 trial. And this was also reflected in the EU Pearl project, because there, for indications, people had the freedom to choose whether to do a platform trial in phase 1 up to phase 4, and most ended up in the phase 2. And I think one explanation could be that in phase 2, still companies are willing to collaborate, but in a critical phase 3 trial, I think people still want to have control over their own trial. So still it seems that the setting for master protocols are mainly the phase 2 arena. And initially, master protocols were applied in the oncology setting. This is also reflected here. And as we have the combination of oncology and phase 2, you can see that the most frequently used endpoint was binary, so it might be response rate. And as far as the use of concurrent control data is concerned, most of them use concurrent control data, and a few share a common control group. And most analyses are still frequentist, but as we will see later on, as some trials can become quite flexible, this might be the time where we will see more Bayesian analysis as well. Good, and as far as the journals are concerned, you can see that still most journals are on the methodological side, and less publications are still on actually published data and trial results. And here you can just see the different sample sizes and blend study arms divided for umbrella, platform, and basket trials. Good. So here a short summary of the main observation of the review. So we can really see an exponential increase of publication over the last year. Still, many are still single arm, phase 2, but as the single arm might be mainly related as they are conducted in oncology in the early phase. And now things might have changed due to the COVID platform trials. Think about recovery or Solid Act, for example. Good. So why are platform trials so different? Here you can see how platform trials look in a nutshell. So they might start as a multi-armed clinical trial where you start with a control group and several arms relating to different treatments. And you might be interested to perform interim analysis to stop early for efficacy, to stop early for futility. So you might close arms early on, but you might also be interested in changing cell location ratio so that more people are randomized to the more promising arms. And this is the main contrast to multi-armed clinical trials. You might add later on additional new treatment arms with new treatments when they become available. So, and in the beginning, you don't know whether one additional, two additional, or even 10, 20 more treatments might enter the platform trials. And as the platform trials are running for a very, very long time, there might be even a change in the control arm over the time. For example, if you see the first treatment arm was very promising, it has very good result, and over the time it might become the new standard treatment, and then it might be not ethical any longer to randomize to the initial control group any longer, and so you might have even to switch the control group, which raises some complexity as far as an analysis is concerned, but I won't touch on this specific topic today. How do you plan such a complex trial design? You have a common master protocol, and then you have sub-protocols for all these different sub-studies. I will come back to this issue a little bit later on. So what are the main potential advantages of platform trials? From the operational point of view, more patients are eligible for a trial due to multiple treatments and substudies with possible different inclusion criteria. And we can use a joint trial infrastructure, which hopefully might lead to savings in time and money in the long run. However, clearly you have to admit that setting up such a platform trial might cost more investment in the very beginning. From the statistical point of view, you can test more hypotheses, which on the other side is also a big challenge. You can share control data, and the adaptive decision rules hopefully lead that we need fewer patients for some of the decision, and it might also allow for a direct comparison between treatments. And you might then also implement adaptive randomization methods where you put more patients into the treatment arms which are more promising, which might be more attractive for patients. However, this comes also with a price as far as the statistical analysis is concerned later on. Good. So where can we find which information? Would you simply write a single study protocol, and whenever a new treatment arm is opened, just add some pages to the study protocol via an amendment? No, this wouldn't make sense because then the study protocol is not readable any longer, because it would include a lot of pages which might relate to treatment arms which are already closed. And for example, if you're a reviewer at an ethics committee or a regulator, you might have to go through a very extensive document, and it's not clear which page is relevant for my assessment. So what is proposed is to have a master protocol which describes the common study elements for all study intervention. But for each sub-study, which we call intervention-specific appendix, you have an own document where you only put the information which is specific to set treatment, and only in combination you get the complete protocol, and you must highlight on which information you can find where, and you really need to assess both protocols to have the full picture, but it helps to avoid to read parts which are not valid any longer. For example, if you have to assess treatment number 2, you don't have to read the parts for treatment number 1. And in a couple of weeks, we will release a set of templates for master protocols, which is based on the Translated template for study protocols, which includes a document for the master protocol itself, a template for the ISAs, but we also developed a DMC chart and a template for the SRP of a master protocol. So just have a look at the UPerl webpage. I think latest at the end of April it should be released. Good. So now let's move to some statistical aspects. Let's have a look at multiplicity issues in platform trials. So what, as we have a lot of non-statistician here in the webinar, usually we use frequentist hypothesis test for decision making in confirmatory clinical trial, where we would like to test the null hypothesis. The new experimental treatment is not superior to control with a frequentist test. There's a famous p-value comes in, and to reject this null hypothesis if the p-value is smaller than 0.05. And this guarantees that the probability of a false positive result, given that the treatment does not work, is lower than 5%. However, we know if we would perform multiple tests with the same threshold, the risk of at least one false positive conclusion increases. Therefore, some smart regulators have pointed at this issue and they say, okay, the control of the study-wise rate of false positive conclusion at an acceptable level alpha is an important principle and is often of great value in the assessment of results of confirmatory clinical trials, as it is stated in the points to consider document issued by the EMA. Good. So now let's have a look at different trial design. Have a closer look and find the difference. Let's start with the design in the middle. So design number 2, where we conduct 2 separate studies. Each study uses a 1-to-1 randomization. And in the first study, we're interested in testing placebo versus drug A. In the second study, we test placebo versus drug B. We have 2 chances for success, but as we have independent studies, there's no adjustment for multiplicity that we conduct 2 studies. Each study can use the full level alpha. That's what we have done for decades, well established. But now look at design number 1. Here we conduct a multi-armed clinical trials with 3 treatment arms. We use a 1-to-1 randomization. Again, we're interested in testing placebo versus drug A, placebo versus drug B. Similar to before, we have 2 chances for success. And traditionally, people would have argued in a multi-armed setting, you have to correct for multiplicity. And as a statistician, a bell would ring and you would say, you have to apply a Dunnett correction for the minimum comparison. Now, look at design number 3. It's now a platform trial where we have 2 independent sub-studies, the 2 ISAs. We use a 1-to-2 randomization in each of the sub-studies, and we test the same hypothesis. We share the control data. Similarly, we have 2 chances for success. But where are we now? Are we closer to design 2, to design 1? Do we need a correction for multiplicity? So what? And if we look more thoroughly on which sources of structural multiplicity we have in platform trials, you will see we might have several sources of multiplicity. Like first, we have several sub-studies with several interventions and different subgroups. Then there might be an adaptive decision-making. We might select some more promising treatments or changing the sample size or changing the allocation ratio. And there might be also several ways on how to utilize the data from the control arm. We might be interested in multiple endpoints, in subgroups. So we have multiple multiplicity issues. And for some issues like adaptations or multiple endpoints, we would treat platform trials not differently from any other adaptive design, and requires that appropriate adjustments are considered. For other sources, it is already well accepted that no adjustment is needed. For example, if in a multi-regional trial, different regulators have different requirements resulting in different statistical analysis plans, it's now well accepted not to adjust. For example, if different endpoints are used in US and by CMA. So what about testing efficacy of different treatments in an adaptive platform trial? So is there a need to adjust for multiplicity in platform trials? So when performing several treatment control comparisons in a platform trial, is the risk really increased? How is the multiple type 1 error affected in a platform trial if a 1-to-1-to-1 randomization is used? If each treatment is compared against the same control group using the full level alpha of 5% for each comparison, then the inflation depends on the number of arms tested in the platform, as shown in the figure number 1 in the excellent paper by Bai. And interestingly, if we look at independent trials, so in each trial a treatment is compared against its own control group, then the multiple type 1 error would be actually higher. So why should one be strict in platform studies, especially if the regulatory risk, the family-wise error rate to improve at least one efficient drug seems not to increase? And such arguments have been stressed by several people. However, some people have also pointed out that due to the correlation, the probability to perform several type 1 error simultaneously increases. And as a side note, this risk can be easily decreased by increasing the control allocation, but then the family-wise error rate would become again similar to the series of independent trials. And here I stole one slide by one of our panelists so this is by Olivier. Due to the shared control data, the test statistic will be positively correlated, and even so that the multiple type 1 error is decreased, some people are afraid that if the control data, if the observed treatment effect of the control arm is on a random low, this means that simultaneously I might reject more experimental treatments for this specific outcome. So this is a trade-off. On the one hand, overall, the family-wise error is reduced due to sharing the control data. However, if I'm in the unlucky situation that my control arm is on a random low, you might reject even more. Good. Let's go on and have a look on what's the combination of testing multiple treatments and performing even adaptations. So what would happen if we ignore multiplicity and that we did adaptation? So first, let's start with a very, very simple design where we just compare one treatment arm against a control group. So this is the column which is labeled here with k equals 1. And for this situation, already Mike Broschen has shown in 1994 that if you perform only one interim analysis, comparing one treatment against the control group and allow for any type of adaptive sample size reassessment, the type 1 error rate might be inflated from 2.5% to already 6.2%. When we analyze the data naively with standard tests without adjusting for adaptation, and if the trial starts with 2 treatments against a control, and you simply select the more promising one at the interim analysis, and you again allow for sample size reassessment, then the type 1 error is already inflated up to 10%. And now it gets even worse. So if you say, okay, again, it's a situation we have 2, but now I select the more promising one, and I allow for an unequal sample size reassessment. So this implies that we change the allocation ratio. So we might increase the sample size for the selected treatment, but we might decrease the sample size for the control group, since the type 1 error is inflated from 2.5 to 70%. And here the clear message is, okay, for the adaptation, you always have to adjust as you would do in any standard trials. So for example, here, if you're interested in sample size reassessment, in treatment selection, and changing the allocation ratio, then you have to apply the adaptive design methodology like proposed by Peter Bauer, Mike Broschan, Chris Jennison and colleagues. So there's no difference to any standard design. So what I'm trying to say here is a master protocol can be an excuse not to adjust for adaptations. You would have to do so like in any standard trial design. However, let's stick to the question if we should adjust for having several treatment arms. And now it seems that more and more people say there's no need to adjust for testing several treatments when they're inferentially independent. What do we mean by inferentially independent? So one argument is that if the treatments were tested in separate trials, no adjustment would be performed either. So why should we do so in a platform trial? And there's been really a controversial discussion on whether an adjustment is needed or not. And there's been a clear shift in the opinions on the need of multiplicity adjustment in multi-arm trials over the last decades. And some authors argue that no adjustment for multiplicity is necessary if independent hypotheses are tested, because if we did separate trials, we would also not adjust for. But the question is really, what do we mean by independence? It's not that the test statistics are uncorrelated. We have seen before, as we use the same control data, by definition, the test statistics are uncorrelated. So this is not meant by independence. It means really that it's not possible that, so we consider hypothesis as inferentially independent if the truth or falsehood of one hypothesis is unrelated to the truth or falsehood of the other hypothesis, so that there's no extrapolation from one hypothesis to the other one. And here you can see a very, very rough guidance on when do we consider it as independent and when it's dependent claims. Like independent claims you have if you're testing different drugs with different mechanism of action. Then there might be no need for multiplicity adjustment at all. However, if you go to the bottom, what do we mean by dependence? If you test different doses of a single drug, because then the hypotheses are related, and then you have to apply a multiplicity correction. So really look on which hypotheses are related, and then you build a family, and only within this family you have to adjust. Good, so it seems now we're ending with a very pragmatic strategy for statistical inference. Each treatment or substudy in the platform trial is considered as an independent substudy, and each controls each type 1 error. And for each substudy, within each substudy, just adjust from multiple endpoints, multiple doses, subgroup analysis, interval analysis, as you would do in a standard design, but you don't adjust for multiplicity across substudies. Good. Another way on how to decide whether multiplicity adjustment is needed could be based on the grounds on what operating heuristics we are mainly interested in. As pointed out here, as far as the power is concerned, we could be interested in just in the per substudy power and the per cohort power, as shown on the left-hand side, or on the power on the platform level. For example, we could look at the disjunctive power, which would be the probability of at least one truly effective cohort being declared successful. And platform trials give now the opportunity to evaluate many other new operating characteristics. You could be interested, for example, in the average power, in time until first success, patients allocated to arms superior to the standard of care. And depending on what is your main criteria, you will end up with a different sample size, and also it will guide you whether you are interested, whether you have to apply multiple testing correction. So like take the COVID platform trials where you compared several treatments, you use a shared control groups. What are you interested in controlling the risk, declaring treatment A better than placebo. Any of ineffective treatments is declared better than control. Several inefficient treatments are approved simultaneously. And depending on what you're interested in, you might be willing to adjust or not. So here, just a summary on how do the objectives of a platform trial determine whether to account for multiplicity. If we are interested in find at least one effective treatment, so one company, a single company is testing several of its compounds, then the control of the probability of at least one false positive decision might be a reasonable strategy. If the objective is to find out for each treatment if it's effective, then an unadjusted analysis might be justifiable. If we're interested in determine all effective treatments, then either do an unadjusted analysis, or you might even use something what is not commonly used in clinical trials. You might aim in calling the false discovery rate. I have a few slides on the full discovery later on. However, before we abandon multiplicity adjustment in these settings, I think it's important to also consider the arguments in favor of addressing multiplicity on a platform trial level. One argument is the current regulatory standard to control the study-wise error rate, which has proven to be very useful in the interpretation of clinical trial results. However, these guidelines might not be directly applicable to platform trials. But we have to be aware that by abandoning multiplicity adjustment relaxes the regulatory standard from a study-wise error rate to a sub-study-wise error rate, which might be okay. Another argument is a societal perspective. If many treatments are tested in an indication, regardless if in separate trials or jointly in the platform trials, we should be concerned about the overall operating characteristics, and it's relevant to assess if treatments are erroneously declared effective, and if so, how many. And platform trials can provide a framework to quantify the risk of false positive. And we can think of different strategy, controlling the formula error at the possibly relaxed level, or considering other error rates, such as the false discovery rate. And finally, an argument is the best use of resources. Sharing control leads to a substantial gain in efficiency compared to separate trials. Instead of spending all the gain in statistical power, you could spend at least some of the gain in the control of the overall false positive rate. So you could argue that the benefit should be shared somehow between the sponsors in terms of power and the regulators in terms of error control. So let me illustrate this with an example. Assume we test K treatments, all with the same effect size. If we perform several trials, each with per group sample size n to achieve a power of 80%, then the total sample size is 2 times n times k. And now assume we spend the same sample size in a multi-arm trial without adjustment for multiplicity, using the optimal allocation of patients to treatment and control. We see that there's a substantial gain in statistical power for each treatment control comparison. So you can see it's the red line. And if we perform the multiple arm trial but adjust for multiple testing, we see that while some of the gain is lost, there's still a substantial gain in efficiency compared to separate trials. This gain could even be further increased if you use a stepwise standard test instead of a single-step procedure, or when we relax the overall significance level. To summarize, I think there's still some room to discuss the requirement of multiplicity adjustment when we make a proper benefit risk assessment and balance the costs of adjustment with the potential gains of robustness of the evidence we generate. So should we use a multiplicity adjustment, but maybe at a higher level? We could debate. Good. So what are the challenges in decision making? As said, the number and type of treatments are not defined upfront. So standard procedures to adjust for multiplicity are not applicable. Therefore, we have to look at online methods, and the online control of the family wise error rate could be quite conservative. And therefore, it might be better to look, if you want to adjust, instead of controlling the family wise error rate, in controlling the false discovery rate. Here I will just give you a catch how it could be done. So it means that traditionally, if you would like to apply a multiple testing procedure, it assumes that all p-values are available at the same time. But for platform trial, that is not true, because this would mean for the first treatment, you have to wait until the very end, since there's no point in conducting a platform trial. And therefore, what is meant by online control is that the hypothesis tests can be made whenever the data become available, but you would like to control the false discovery rate at each step. And at each step, a decision has to be made if the current null hypothesis should be rejected based on only the previous decisions. And for many online procedures, they only work for independent p-values, but in platform trials, we have positively correlated test statistics, and there's some work on this topic. So how can you use such new methods in platform trials? It's simply the combination of traditional methods where you would adjust on the one hand for testing several treatments, and you use other methods like for testing the hypothesis also at interim analysis. And at the end of the day, what you really would like to control is the expected proportion of treatments that are falsely declared efficacious among all treatments that are efficacious. And here, so we did it in the context of platform trial. The purple line is for the setup where we test 10 treatment arms against the common control. And the purple line is where we would do no adjustment. The yellow one where we would do a Bonferroni adjustment. And you can see with the online methods, you are less conservative, like the Bonferroni. However, we don't gain the full power like where we would not adjust for all. But still, I think the false discovery rate could be a very valuable mean in the context of exploratory trials, platform trials of a single company where you would like to test as many compounds as possible. Good. So let me summarize some multiplicity discussion in platform trials. So we said whether you should adjust or not depends on whether your claims are independent. So think about this scenario. We have different treatments with different mechanisms of action and same control. It was one of the questions we would forward to you in the survey. And here, I think many people would now argue, no, there's no need to adjust for multiple comparison, because for sponsor 4, it doesn't matter whether the hypothesis for sponsor 1 is rejected. So in this setting, no need for adjustment. And also here, where the control data are not shared, no one would adjust for multiplicity, because here the picture becomes even clearer, because if this sub-studies were set up as completely independent trials, no one would adjust for multiplicity, so no need to adjust here. But now look here. Now we have again a common control, but now we have 3 arms for drug A because we have a low dose, medium dose, high dose, and similarly for drug B, we have a low dose, medium dose, and a high dose. Would you adjust for multiplicity? No adjustment at all. Adjust for the total number of treatment arms. So we have 6 treatment arms, or you say, okay, this is a joint family for drug R, so I will only adjust within the sub-study for drug A, and similarly, I would only adjust within the sub-study for drug B, but I would not, the adjustment for drug A is not affected on how many doses are tested by B, because these are completely independent families. So really, this is an example where we have one claim, some claims which are independent for drug A and B, so we would not adjust that we are looking at 2 drugs at the same time. However, within each drug, we have to adjust as several doses are tested. So the concept of study-wise step-on rate is not directly applicable to platform trials, especially if they are perpetual in nature, and the control of the family-wise error rate at treatment or substudy level seems to be a pragmatic approach. But do we really have a consensus on what we have to consider as independent? And also the overall operating characteristics of platform trials are important, depending on the trial objective, the control of the false discovery rate or the family-wise error rate are a possible option. And also other sources of multiplicities have to be taken into account on which adjustment makes sense. And as said before, when we speak about adaptations, there you would have to use the same methodology as in traditional trials. Good. So now let's move on to the next big block. Which control data can we use? So we have fewer control patients due to sharing controls. So here you can see classical development for K treatments, and we have K separate trials with a one-to-one randomization, and the sample size to reach pairwise comparison is with a bar of 80%. And then we compare to a multi-arm trial where we use an optimal allocation ratio of 1:1:1 to square root of K, minimizing the overall sample size. Again, we want to have the same power, and you can see overall we save a lot of patients in the platform trials because we need less control data. So we have a fundamental save of patients required for the platform trial, especially if the number of experimental arms increases. And similarly, if you just look at the control data, and this might be of special interest in rare diseases, that you don't need so much patients in the control data, but still you can have some control data. Now the question becomes, can we use all control data which is already available? Because look at this situation. So the platform trial starts with the control and treatment A, and only later on treatment B is added. And now the question becomes, which data can we use for the comparison of B versus control? Can we only use the concurrent controls for treatment B, meaning patients which were randomized at the same time? Or can we also use non-concurrent control data for the comparison of treatment B to control? On the one hand, if we use all control data, we increase efficiency. So it's easier for us to reject the null hypothesis. However, if we use non-concurrent control data, this may introduce bias due to different type of time trends. Are non-concurrent controls, is it the same issue that as we discussed about using historical controls in randomized controlled clinical trials? Non-concurrent and historical controls share several sources of potential bias, And when using historical data for comparison in clinical trials, we accept that the strict type vulnerable control is not possible. So what about platform trials? Non-concurrent controls have a much higher standard because they're collected within a framework which has many features standardized. So we have a much better data quality. Same infrastructure, assessment of endpoints, monitoring, all changes are well documented, and patients are still randomized, and blinding is possible. However, non-concurrent control data also have been randomized and were blinded, but at different calendar time, such that randomization does not ensure control on the distribution of prognostic factors between the non-concurrent control data and experimental arms, and patients and investigators are not blinded with respect to the experimental treatment and the non-concurrent control it is compared to. So there's a lack of true randomization, which can induce time trends. And there are time trends due to external and internal factors. For example, externals, the changes in standard of care, patient population, especially in the COVID trials. You have seen there were several waves in the pandemic, and the data are not really comparable in the different phases. But there might also be some internal time trends, like change in recruiting centers, and an analysis stratified by center is not longer possible if center enter and leave the platform trial. There might be a change in recruitment strategy if promising treatments enter the platform trial. There might be a change in inclusion and the exclusion criteria because of other experimental treatments. And there might be a change in assessment of endpoints if new diagnostic devices become available, because platform trial might run for a very, very long time. Good. Despite this, if we come to the conclusions that we would like to perform an analysis also using non-concurrent control data, what are natural approaches? One could be completely ignore them, which we call a separate approach, which means to be on the safe side, we use only the concurrent control data. Or you might do a very naive approach where you simply pool all the data and you analyze. Your analysis is based on concurrent and non-concurrent controls. Just smash everything in. Or you could think about a model-based approach, which adjusts for time trends by including time as a covariate in a regression model, for example. So here you can see how it would be done. It would mean that we would have a standard effect for the treatment effect as we would usually have. But whenever there is a change in the platform trial, like change of allocation ratio, a treatment arm is leaving the trial, or a treatment arm is entering the trial, we would define a period. And this period effect is adjusted for in the statistical analysis. What are the underlying assumptions and properties of such test? For platform trials without interim analysis or other interactive elements, this model-based approach leads to a valid treatment effect estimator regardless of the functional form of the time trend, if the time trend is the same in all treatments, and if the time trend are additive on the model scale. And here you can see the results, and you can see we cannot simply pull the data and do a conventional analysis, because regulators might become quite nervous. So this is the blue line, the pooled analysis, because here, depending on which time trend you have, you might see a huge inflation of the type vulnerability rate. However, the regression-based model, it controls the type vulnerability rate. When we compare, for example, two treatment arms against the control, and on the x-axis we see the strength of the time trend. So you can see the green, we have separate analysis, and the regression-based model both controls that vulnerability, but here, when we simply pull the data, we would have inflation. And this is shown in the paper by Marta Profil, that you get an unbiased treatment effect regardless of the functional form of the time trend, if the time trend is the same in all groups and the time trends are additive. And when we use this non-concurrent control data, you can see that compared to the separate analysis, we really have an increase in power, which is the good news. However, the bad news is if the time trends would differ between treatment arms, the estimates are biased, and then also for the regression model, the type 1 error might be inflated. So this only holds if you are willing to assume that the time trends are similar in all groups. And I really don't know how realistic it would be to assume that the time trends should differ between treatment arms. So here you can see the reference to the paper where there's a more in-depth discussion. Clearly, there are much more methods, also some nice Bayesian methods. And here, if you are not on Twitter, you might take your chance and go on Twitter for the first time, because there's a nice tutorial by Gerd Viele who discusses all these different methods on how to incorporate non-concurrent control data in his tweets. And in addition to using frequentist model-based approaches, you might have a look at Bayesian time machines, which is coming out of the Paris, or you might even use techniques well known in the area of meta-analysis. You can also use network meta-analysis to combine the different pieces of the times where you before and after a trial enters the platform trials. Good. Good. Usually, you know the previous control data. So what if previous control data is known when new treatments enter the platform trial? So we would have the appropriate methodology, but if arms have already left the platform and they are published, the outcome data for the respective control group is out in the public domain, so it's known. And a platform trial with a control with random low in the outcome can be an incentive for sponsors to join the platform and to plan an analysis including non-concurrent controls. And conversely, a platform trial with a control with random high can be deterrent to join the platform trial, and you might also not go for an analysis incorporating the non-concurrent control data. However, if a sponsor would act in such a natural way, you introduce bias and your results are not any longer valid, even if you go for a model-based approach. So I don't know how we can avoid such strategies. And by the way, do you think that we can always use all concurrent control data? Because in the beginning I said, okay, the sub-studies might have slightly different inclusion and exclusion criteria. And would you use control data which wouldn't exactly fit to the sub-study concerned? And in platform trials, you might give the patients the choice to decide to which treatments he wants to randomize. Again, if a patient is only randomized to a subset of treatments, would you then use all concurrent control data? Because this might include control data of patients who could not be randomized to the treatment group of interest. So even for the concurrent control data, a principle would be that you can only use the control data of a patients who in principle could have been randomized to the treatment group of interest. Good. So let me give you a summary on non-concurrent controls. The inclusion of non-concurrent controls is a question of variance bias trade-off. Methods to address potential bias are available. However, speak to your statistician because they rely on very specific assumptions and they are not always obvious. And the problem of pre-specification is difficult to address. keeping control data blinded may not be possible if treatment arms are stopped and results are reported. And in broader indications, regulators might be very reluctant to accept analysis using, I can hear something. And in broader indications, regulators might be very reluctant to accept analysis using non-concurrent control data as well. But I think in rare diseases, the situation is different. And in rare diseases, we see still a lot of single-arm trials. And I think anything which incorporates at least some concurrent, but also non-concurrent control data must have a higher level of evidence. So I think this is the field where we can explore the use of non-concurrent control data as well. And if non-concurrent control data is utilized as primary analysis strategy, I think it's still reasonable to also present the analysis using only the concurrent control data, but possibly with a more relaxed significance level. Good. Dieter, how much time do we have? Do we still have 5 or 10 minutes?
Yes, of course. I think you can proceed.
Okay. Then just a few words on the role of clinical trial simulations, because traditionally, when we speak of designing a study and which sample size would be suitable, we would just plug in our assumptions into the t-test or chi-square test, and we would come up with a sample size. So quite easy. However, now platform trials are quite complex. We have all these possible adaptations, and we even don't know which treatments will be tested eventually, which means analytic solutions are often not available. So we need clinical trial simulations to evaluate the operating characteristics of a platform trials. It means that we try to simulate realistic platform trial trajectories, where we have to make a priori assumption on the timing of analysis, allocation ratios, when will be new treatment arms available, and we do it again and again, and we then look what would the power, the average power, and so on look like. However, how should you start with? And here we propose a very, very simple concept which you call the vanilla sprinkles approach, which means start with a very, very simple design. So the vanilla design provides space. It's a very, very simple design. And then you add sprinkles like the impact of interim analysis, the impact of using adaptive sample size strategy, or change of allocation ratio. And you do it step by step, because if you start with the most complex one, you don't know whether the adaptation is a good one or not. So start very simple, and it's really an iterative process which takes a while. So setting up a platform trial, it can take long, because different people might have different interest and different object. So one would ask, can we stop earlier? Some will say, okay, what if the group is faster? So you will start with a very basic design. You will do your simulations. You will discuss your results with different stakeholders, with patients, with clinicians, with regulators, and then you will adapt your design. And like in EU Pearl, some discussions even took 1.5 years until you agree on a common design. So setting up such a platform trial takes much longer, and the simulation can become quite tricky. And then you might ask, okay, which software can we use? So we did a review, and there's not much software out. You can find, you can use some software initially made for multi-armed clinical trials and play around a little bit, but usually you would need tailored code. And here on the right-hand side, you can find which code we have developed, so for the different indications, and based on the learnings for very specific designs, we tried to create a very simple platform trial simulator, which is quite generic, which should help in future to adapt it more easily so that you don't have to start from scratch every time. Good. Now I come to my conclusions. So hopefully I convinced you that sometimes a platform trial or master protocols might be a better use of resources. There are operational and statistical advantages. However, they're also challenging. And don't be scared. In master protocols, it's not necessary to adjust for all potential sources of multiplicity. Sometimes you have to go for a very pragmatic approach. But as far as the analysis is concerned, be transparent when you use non-concurrent control data. I think in rare diseases, it's reasonable to enrich your analysis by non-concurrent controls. However, be transparent on what are the assumptions what you use, and this implies also early discussions with regulators to see and learn early whether they would accept such type of analysis. And you really have to use very tailored methodology to improve the efficiency of master protocols, and start simple and don't try to do all the adaptations, selections, interim analysis at once. Start step by step. So that's more or less all I wanted to say.